Hello.
I have to get myself an ACSA shirt. I'm still in my work clothes.
Oh, yeah, I've got to do that next. Thank you for reminding me. Can you hear me okay?
Yeah.
Oh, yeah, that's better. I couldn't hear you before because David has the sound right down. I shall be back. I have, I don't have any pictures to show you because I don't know. David's got this set up on his work. That's why I come up as David Manning because I'm on his laptop. But I shall go and get my T-shirt on, but no one will see me if I don't have a video.
Yeah.
How do I start video? Oh, hang on. It goes. Does that do
it's done something.
Oh, yeah, that does something. Yeah.
Are you able to
hang
lower
on.
your
Wait, because David's really tall, so it's like up there, and I'm like getting a sore neck now, but that's better. Yeah, so the only thing I can do, I don't know, David, how do I change the name on there? Because it's on your thing.
Well,
I've got to go and get my
while you're doing that, Louise,
yeah.
Shall we see if it looks like Helen's having trouble joining, because it's Sandra and Helen who are speaking tonight. So shall we check if they're audio and able to share screen and everything like that?
I'll go get my shirt on. And David's here. Wait, please, honey.
Okay. So
my lighting is pretty crap.
Shows mine like light in the face.
I have to hold my lamp and cross my head. I'll be like,
yeah, that's right.
Oh, no.
You can adjust some of the lighting in it, but anyway.
Is that all right, or is that too shitty?
That's. I think we can come back to that, like Janine was saying. It looks all right.
Hello, Helen. Looks like you're finally been able to join in the meeting. Taking.
Now, how do you do the back? Hey, do you guys know how to do the background thing? I haven't ever done that before.
You know where it says stop video? There's a little button next to that.
Yeah. Yep.
There's video settings.
Choose virtual background.
That's right. And there's a little plus. Yeah, backgrounds and filters, and there should be a little plus there, and you can add an image. So Janine sent out that image just before.
Yes.
I can put it in. Yeah. And there's also an option in there to touch up your appearance, which I like to put at maximum and adjust. No, just me. That's just me and the students, you know.
That's hilarious. Where's that setting?
It's under video, my video.
That's
so that you look like a bit... Yeah, it's just does that kind of 80s soft focus, I think.
I want that because I'm pretty sharp with my light now.
Yeah, that's it. And there's but you can also adjust for low light. That's really what I was going to say.
I have to find the.
Can I just before we get too stuck into all these bits and pieces, can I just check that it gave you all a warning that we're being recorded?
Yes.
Yes.
Okay. Great. So it should record to the cloud, and then I'll send you the link.
So we're not able to mute noisy people or anything like that, are we? We don't have any of those sort of controls.
Let's see if you can mute. Can you mute somebody other than me? Can you mute each other?
No.
Can I just interrupt then every time and say, could you please, I'll just tell everyone and ask everyone and take a pause to make sure that everyone's muted. I'll just be very direct. And if there's any noises, I'll make sure to tell people off.
Yeah.
I'll just be diligent at that. All right, background.
How are you, Helen?
I'm okay, yeah, not bad.
Good.
Just, aged mother problems really hit home today in a big way, which has thrown me a bit in terms of prep and other stuff.
Oh, shame.
Yeah, okay.
She's just gone backwards at a rate of knots. Anyway,
Helen.
Sorry to hear that, Helen.
Oh, well, she's 95. Like, where else can she go?
Wow.
You just want a simple, fast passing for them at this stage, because she's not enjoying life. And I have to share this bit. They gave her a catheter for a whole week and sent her home.
Wow.
For us to look after. My brother principally.
Wow.
Oh, happy. Anyway, so the joys of looking after your mother at home.
Very difficult for her dignity, isn't it?
Hmm?
Very difficult for her dignity.
It's only difficult because they overreacted to something that they thought they saw instead of actually teaching her how to avoid. Anyway, sorry, I just have to get rid of a bit of.
No, it's
Yeah, that's fine.
yeah.
Do you want me to share my screen at some point, or have
Yes.
Yeah,
Yes,
let's
please.
test that.
you try that?
And also, are you able to I don't know, you seem very quiet, soft.
Let me
Is
see
the
if I-
audio?
Oh, I haven't got my audio up. Is that better?
I think we will be able to hear you, but we'll just have to make sure that everybody mutes. I just found a button that I can press. I can come in between cooking rice and getting my son in and out of the shower, and ask all to unmute. I'll ask all to. Oh, I can mute everybody, but then that would bugger you around though. That's the problem. It would stop. It would also mute.
So you can mute everybody as long as we're all comfortable knowing that we need to unmute ourselves. Is that?
Yeah, but that'll include Helen. Like, I think that you can, yeah. I think I have set it up so that when you come in, all right, yeah, I'll mute everybody before. Let me do it now. Okay, mute all current and new participants and allow participants to unmute themselves. So you should all be muted now. And
yep, we got muted.
Can you? Okay, so you can unmute yourselves. Okay, so I'll just come in and do that every now and then, unless Helen's speaking.
Yeah. Thank you. Oh yeah.
Yay.
Beautiful.
Great.
Helen, what are you referring to your project as? So if we are telling people, if you want to know more about Helen's such and such project, what are you calling it?
Mute Helen.
Yes. Helen, we
Hi,
can't
Michelle.
hear you. You'll have to unmute. I can probably
My internet
ask you to unmute.
connection is unstable.
Oh, it looks like I can make you a host.
Yeah, you should be able to make others hosts as well. You can make all of us hosts, and that way we can do things in the background.
Yes, I wasn't able to set it up that way because QUT required a QUT email, but I've just found another button. So I'm making you all co-hosts.
Okay.
That's right.
With that co-host, I might be able to live stream it that way. We'll see.
Do we want to live stream?
Yeah.
If not, I'll just record it on my computer as well, if that's okay, Jen, and then I'll upload it to YouTube and Facebook later.
Yeah, okay. If you don't think that will be an issue with stuff, that's fine with me. It's currently set up to record to the cloud. So just for what it's worth. So you're just going to do a screen grab, are you? Or are you going to change the Zoom settings? I don't mind, but it might stuff it around, but it's you sound like you know what you're doing. So that
I
works.
don't, which is why I was going to come in and like try it first.
Okay, all right.
So while you're
I'm just trying to
trying
figure
that,
out where it is.
while you're trying that, can we now turn on the waiting room, seeing as we can let people in rather than have to jump in while we're still sorting ourselves out?
I don't know if we can. I'll see if I can do that.
They usually don't allow it until 15 minutes before, and we've got another 3 minutes till that hits.
No, the meeting time, I had to set it to 6:40. So even though they don't know that, it is actually open now.
Thank you so much for doing all of this, Jen.
Oh, this is
Yeah, that's
nothing. You
such great
guys do it
help.
all. You guys doing it all. I'm not doing anything. This is amazing.
No, that would have been an extra huge big thing for us to do, I think. Yeah, no, it's good that it's really lovely because everyone's kind of just pitching in. Thanks so much, guys. It's so awesome.
Oh,
That's
it's such
really true, actually. It's been really collaborative. It's nice. Good.
Yeah.
And thank you, Helen. I really can't
Yes,
wait
thank
to
you,
hear-
Helen.
Yeah.
to hear your presentation.
I can't see Helen. Oh, because she's
Can't
on
see
the.
you here, Helen. So I've just asked Helen to unmute. I wonder,
Am I
there
muted?
you
Is
are.
that better?
That's better.
You
I
just
can't
have your
see myself.
no. You just need to turn your video on.
Sorry?
Ask to start video. Are you able to start your video now? There you are.
Yay, there we are.
Can you see me?
Yes, we
Yeah.
can see you.
Because
Great.
while I've got the slides up, I can't see myself at all. So just-
Can I sit? Yeah, sorry, go on.
go on.
Oh, I was just thinking, I don't know about you guys, and do you want to ask people to turn their videos off while Helen's speaking, or just leave it, just mics, eh?
We should be able to do that if are we all?
Yeah, you're all
All
co-hosts
hosts.
now. So I can stop
Doesn't
Janine's
say
video. Oh,
Does it affect bandwidth? Like, does it affect the
Yes.
Zoom meeting when everyone's
yeah.
got? Yeah,
that's
I
what
would
was
prefer that
thinking.
because then, yeah, because I do always worry about my connection.
Yes.
So, yeah, I think Helen's pretty good knowing that we're there without, you're not really worried with the gonna see
I
us.
don't
Hey,
need to-
Helen, you're more like
I don't
looking.
just-
Yeah.
Can you hear me?
Yes.
Yep.
Okay. I don't need to see people. I just need somebody to tell me I'm presenting, looking okay.
So at the
Yeah,
moment, you're on
I'll
the
do
slide, you're
that.
kind of, I can only see half
Sorry?
your face.
Sorry?
Oh, yeah.
Yeah. So Helen, you're going to have to move your camera. You have to poke it a bit more towards your nose, because at the moment we're
To your
seeing your
left.
left ear. To your right.
Move towards your left. Oh.
I can't even see what I'm doing, and you ask me to do things in mirror. So
Yeah.
have I moved it the right way?
No, keep going. Yep, bit further.
Uh-huh.
It's just bouncing back to that spot.
Yeah, it's bouncing back to that spot. So you want it to come further towards your right and down slightly.
Okay, I have to just shift the whole thing across. Sorry, I'm on a tablet. It works.
Okay.
That's work. That's
That's
almost,
works.
Okay.
it's just clipping your ear, but we can see your whole face now.
I'll try and put something behind it. No, that's funny because it looked exactly right when I looked in the camera earlier.
Maybe
Probably
if we
because you were
sit
staring at
backwards.
it.
Yeah.
Probably because you were in front of it staring at it. And then you moved. You know what I mean?
So is it okay now,
Yes.
or is it still clipping my ear?
It's
You're-
you've got about this much.
Yeah.
I can't see a single thing. So
Imagine that
sure.
your screen is divided in two vertically. You're on one side of it.
yeah.
Okay, you're on the right. You're on the left side of it when I'm looking at you. So we
Yeah.
can see you. You got a
Yeah,
nice
that's
colourful
good.
thing in the background.
So if I move across like this, I'm fine.
Yes.
Yeah,
Yeah,
what-
if you move left to your
What's
left.
the
yeah.
brown thing behind you? Yes, with what you're doing. Oh, I just
Sorry,
confused the
that's my-
situation. Sorry.
That's my
No, I
problem.
was suggesting you
that's-
move closer to it.
Sorry.
That's okay. Don't worry. I just confused you. You were doing fine before.
No,
Is
that's
it-
good. Yeah, the fabric behind you is lovely. So we can see that very well. Yeah, that's side.
Excellent.
Hi, Maxine. How you doing?
Good.
Um,
Should I speak or type?
First of all, you can try putting your camera up a little bit, because at the moment all we can see is your boobs.
Oh, I couldn't even see me. My camera.
Oh, there you go.
My camera's very dodgy. It could disappear completely at any time.
Hello,
Oh.
everybody. Thanks for joining in. We're just doing some last minute setup.
Oh, there's trying to see my boobies again.
how to put the sexy into citizen science.
Exactly.
Hubba.
Oh, dear.
Yeah, see,
We
now I
need those
froze them.
socks.
Oh yes. Hey,
Froze them.
Hey,
just
Janine.
quickly, Sandra.
Oh.
I want to just mention that if you want to reduce your bandwidth consumption, under video, where you had the adjust for low light button, there's also an option to switch off HD on your video, because it's doing a default now where it's defaults to high definition images and video. So for zooming from home, I find that really useful.
Awesome.
How do I change it? Because it does say FaceTime HD camera. How do I reduce the camera
underneath that, it'll say original ratio and then HD. Do you see that? Just untick the box that says HD.
No.
I've probably gone.
Do I have to go to video settings maybe first?
No, it sounds like you're in the right
Thank you very much.
place.
Yep. Got it. I got it.
Yeah. Okay.
And mirror my video. Do you mirror it? No. I don't know.
Depends
Anyway.
on whether I want to
Got
know
it. I
which
got it.
ear
Thank
I'm
you very much.
scratching.
All right, I'm going to send out a reminder that this is on our Facebook channel, YouTube, Twitter, etc. We ready?
Thank you.
Can somebody check me again, because I just realised I had my thing around the wrong way.
I think you're looking good. Now you're on the other side a bit too much. You've moved to your left too much. Oh.
Yeah, no, that's terrible. Is that better?
Oh yeah, you're kind of in the middle now. Yeah, it's good. Looking good, Helen.
Helen, for what it's worth, at the top there's a view options next to the green thing that says, and if you click on that,
I'm on a tablet and none of the instructions.
Okay, all right.
But I've tried it before.
Okay,
I know this.
all right.
I know this works. I actually don't do Zoom very often, but I know this particular combination with my old technology works for doing a presentation. So I've stuck with it.
So you guys are able to mute each other then?
Yeah.
Okay.
Let me just do
How do you do that?
Jennifer, I think you've got us all. Sorted.
Do you just
Yeah,
go
it
to
sounds that way.
participants and you mute them? Oh no, that's invite.
Yeah, you can just so you hover over the picture of somebody on the right, and then you get three dots, and then you can see the button mute.
I can. Yep. Thank you. That's so cool. I didn't know all this stuff.
And I think we can make see a spotlight for everyone button there. So maybe we can make Helen the spotlight. Oh, there we go. I've put Helen as a spotlight. I don't know if you want to do that. She's gone.
It's gone.
Helen's disappeared.
You did a magic trick and she's disappeared.
And now I've lost my gallery view. Now I've lost my gallery view. Damn it.
Uh-oh.
All of this is being recorded, isn't it? It's all the daftness.
Oh dear.
There we go. It's under meeting.
It'll get edited out.
Thank you, Michelle.
Just cut it all out.
And yet we can still see
Editing
Helen,
is my friend.
and yet we can still see Helen's screen.
And that's
yeah,
great.
she's coming back one day.
Oh, there she is.
Yeah.
Oh, you mean she's walked out? Oh, okay.
Yeah. She's
Okay, so I'm going to suggest, just to conserve bandwidth, that when we do start, we'll make the introductions. Janine and Sandra, which one of you guys are going to introduce Helen in a few minutes?
I'm going to have a go. Yeah.
Going to have a go. Excellent.
Have a go.
Janine, you might want to start with a welcome to country.
Sandra's going to do all of that.
You do all that? Okay, cool.
Sandra's hosting tonight.
Excellent. Fabulous. Okay. So Sandra's
No,
hosting.
Is there anything else Sandra needs to know?
I might ask Janine for some help.
So Sandra, I would suggest you
I've
talk about
got-
some housekeeping.
Mhm.
In other words, tell people to turn their mics off, and to save bandwidth, also turn off their video, their particular video stream from them.
Excellent.
Okay.
Thank you very
So that's
much.
the best way to go about it.
Yeah.
Let them know to drop anything in the chat We will be going through and answering questions in there whenever we can. We'll also be, Janine, myself and Louise and whoever else will be going through and double checking at the end that every question has been answered. If we don't get to some of them, we will send them to Helen at the end, I believe. Is that correct, Janine?
That's
Janine's
fine.
nodding at me. Excellent. Awesome.
And I've asked Helen if she could be contacted via her Facebook page as well, because that she does
Okay,
have
so Helen, do you want to just
one
flick that
and
Facebook page at me at some stage today, or just stick it in the
probably
chat?
Oh,
we can-
sorry. The technology for that just escapes me.
Okay, so what are you called on Facebook?
Just my full name with spelt CKE.
CKE.
But I've got a Inviting Nature to Dinner page, a Bring Back Our Butterflies and Other Invertebrates page, an Australian Native Bees page, Earthling Enterprises page.
Okay, so
was
we're,
good.
Oh wow, I didn't know that.
Oh, well, that was
as we're talking about the Bring Back the Butterflies, I will hit that one. Okay,
yeah.
I will
that's
share
it.
that one.
Inviting Nature to Dinner is probably where I've moved to the most, but I'm not a big Facebook user. I just answer other people's stuff.
Yeah.
So, but I've got it all on a slide at the end anyway.
That's okay. I'll just keep going and putting them in the chat as we go, because that makes it easier then.
My website's probably, I know a lot of people, yes, Lou's website's less, www.earthling.com.au.
Okay.
Gives most people access to most things. So.
All right, I'm just opening that up now so I can start tagging you on things. Now, are you on Twitter or anything like that? Twitter,
Yes,
Instagram?
No, I just, sorry. I'm an early adopter, but still old school.
That's okay. That's fine. Just in case I had to tag anything, that's all.
I am on LinkedIn, so that's a possibility.
Okay.
I spend too much time behind a computer already. But yeah, so yeah, Facebook and website are the main sources.
Can I just add something that might be useful for you guys if you don't already know this? People who are
Can I ask
to
a
the
question?
meeting.
Do you
Hang on,
want
Louise,
the
just a sec.
members?
Just a minute, Louise.
Just a sec.
Hang on, Louise. Yeah.
People who are new to the meeting and are new to the chat won't see old chat messages. So it might be a good idea to sporadically keep posting if more people are joining that we'd appreciate mics being muted. And like you said, Michelle, that all the questions that are in the chat, we'll make sure that there's time to answer them at the end or in another session.
Yep. I will
So
put a little, I'll put a little blurb.
Yeah, and just copy and
blurb as I
paste
go.
it in every few, like every 10 minutes, or if more people are coming, you can see if they're
Just a
coming. I
Yep.
see we have got people joining us now.
People are starting. Yep.
Yeah.
I just wanted to ask Michelle one question. Michelle, did you want us to stop our audio recording as well and just have no recording, or what? What did you want us to do for us? Because you said something before, you'll ask all the participants to take their audio off.
Yeah, so Louise, I would say we'll
Mute.
ask everyone to mute themselves.
Yeah, but what about
Turn
video?
off their video, just so they can see the audio visual better.
Yeah, okay.
And then I would suggest we do that as well ourselves, but except for Helen and maybe Sandra.
All right. So mute our voices and take our audio off. That's what you'd like us to do?
Yeah, no. Mute, stop video and mute.
Stop video and mute. Okay.
Yeah. So there should be something somewhere that says stop video. That's them seeing us type video, not them seeing Helen. Okay.
Yeah.
So yeah.
Yeah, that's them.
It's our camera.
Yes, got it.
Yeah, so it's
Yeah.
basically like turning your camera off pretty much, so
Yeah.
that you're no longer visible to everyone,
Yeah.
which
Just
just
like
saves
that.
us bandwidth.
Yeah.
Yeah, you've disappeared.
Yeah. And my name will stay there then. Okay,
Yes.
good.
Yeah.
All right.
Yeah.
So we're letting people in now?
Yep, everyone's coming in. Yep.
Good.
Welcome everyone. 24 people. Welcome. Please feel free to. We're already here. Yeah, we know, Angela. It's okay. We're just chatting. Please feel free to introduce yourself in the chat and tell us where you're from.
Will someone record the chat?
Yeah, I'll keep saving as I go.
I don't have access, so I would really like to see the chat.
Yep, cool.
Yeah.
So it's 7 o'clock. Should we give it a go now? Should we start?
I would give another 5 minutes just to let everyone get in. Oh, Liverpool,
Okay.
UK. G'day from Australia, Liverpool, UK. Maxine, North Coast. Tessa and Joe from Springwood in the Blue Mountains. Claudia in Canberra. Hi, Claudia. And Ellie from Brisbane. Ian from Gympie. We got people from everywhere. Gubby Gubby, excellent. Awesome. Love a good Gubby Gubby country. Eaton's Hill. Rose Astonville. Where is Rose Astonville?
Wow, Melbourne.
Northern New South Wales.
Northern New South Wales. Okay, cool. Then from Tamworth. Northern New South Wales.
Woohoo! Sorry. Hi, Ange. I know Ange.
Let me put mine in.
Should we put my, yeah, let's all put ours in too. We're in Waka Waka country, I think, Ange.
Oh. Kondamuka here. Westlake. Go Westies.
Hi, Ed. The only name I've managed to catch so far that I know. I know a few.
So while we're waiting, we'll probably just go around and introduce ourselves, starting with our chapter chair from Queensland, Janine.
Thanks, Shell. Hello, my name's Janine Bedros. I am the chair of the Queensland chapter of the Australian Citizen Science Association. It's very nice to see you all here, and I'm thrilled that we've got people not just from Queensland, all around Australia and all around the world. I've been involved in citizen science projects for about 10 years, and I just love it. So nice to have you here.
I'll probably go next. My name is Michelle Neal. I'm the social media coordinator for the Australian Citizen Science Association and a member of the Queensland chapter because I live in Queensland. So g'day everyone. Lovely to see you all here. Louise, do you want to introduce yourself? You'll have to turn your mic on. Mic on.
Sorry. I'm Louise. I'm the secretary for ACSA Queensland. So hello, and I'm from Gurrapul Yagara country that's in Ipswich.
I'm Jen. I'm a committee member and also from Turrbal Yugara country. It's so good to see you all here. It's very exciting.
Is that all of us from ACSA? Yeah, so I'll introduce myself. Yeah, it's my turn. Hi, everyone. Welcome. Thank you so much for coming. My name is Sandra Tushinska, and I am an avid citizen scientist lover and citizen science appreciator. And yeah, welcome. Thank you so much for coming. And we have Helen today. So I'm sure you all know that already. But I'd like to, first of all, just do a little bit of housekeeping for all of us, if that's all right. So I'd like us all to mute ourselves when Helen's speaking and also turn your video off so that we can actually have more bandwidth for the video, because as you know, sometimes it's hard doing these things online with many people. So that will be the one thing that we would really appreciate all of us doing. And also drop your questions in the chat section. Helen will try to answer them in the Q&A at the end. So please feel free to use that chat. That will be monitored over time. And before we start, I'd really like to acknowledge the traditional owners of the land on which we are all currently on and pay our respects through the desire to learn from our traditional elders and those emerging as well. And for me, it's a personal thing about learning to better understand how we can be in a prosperous relationship with this amazing landscape that is Australia and its people. So I'd like it to make it a very personal acknowledgement of country, because I feel like the more connection we have to the land and to each other, that's the ethos of doing the acknowledgement. So to introduce also ACSA. You may or may not know what it is, but it is Australian Citizen Science Association, and it's member-based and incorporated association that seeks to advance citizen science through sharing of knowledge, collaboration, and capacity building and advocacy. Does anyone know, maybe I should say, what citizen science is? It actually involves public participation and collaboration in scientific research with the aim to increase scientific knowledge. And this means that the public can be involved, which is so cool. And scientists are also involved a lot of the times to lead projects and to be on an advisory committee for those projects. But basically, we want to collect and analyze scientific data in relation to the natural world, and predominantly collected through citizens like you and I, or any other person that likes to join these projects. And as I said, scientists and field experts are often involved. And so our goal really in ACSA Queensland is to build awareness of what ACSA does and the citizen science community that we have, and the opportunity to get involved in science as much as we can. So without further ado, I'd like to introduce Helen Schwencke, who's right there. If you can see her, wave, Helen. But Helen and I have a long history now, I would say. We both share a huge passion for the little creatures. And Helen has been a tremendous influence on me in trying to understand the relationships that they have with their host plants. So Helen has been actively involved in looking at the intricate relationships, particularly of butterflies, raising their caterpillars and understanding what plants they feed on. But it goes so much deeper. Helen's got a huge wealth of knowledge on this subject. She has even started the Butterflies and Invertebrates Club in 1994 in Brisbane. And, you know, she's got a garden at home that's wealthy of in wealth of these amazing creatures. She's been raising them and creating awareness and sharing that knowledge. And today we're lucky enough to have her share that knowledge with us. So I will let Helen speak for herself now. Thank you so much. And I hope you enjoy this presentation.
And thank you, Sandra, and ACSA, sorry, Australian Citizen Science Association, Queensland branch. And yeah, and I'd really like to acknowledge the Turrbal people. The more I do this work, the more I realise that there's just so much more depth in absolutely everything, and that we're just scratching the surface. And to be able to, yeah, just understand so much more. So I respect their elders, past, present, and emerging. So my story is of a journey that, through what would seem like a lot of random observations, that have led to deeper understanding of ecological relationships, and there's the learnings that I've gathered along the way that I hope will help inform citizen science practice into the future. So each of us has an anchor point for whatever path we choose, whether we're conscious of it or not. And in my case, I was probably not yet 2 years old. And it can only have been out at Indooroopilly, which was then the frontier of Brisbane's urbanisation. My newly migrated parents had bought 24 perches, I don't even know how many square metres that is, of extra airy farmland. Some of the trees had been removed, some of the shrubs had been removed, but they were building a family home in 1955. It was before the time of improved pastures and before all the invasive ground covers that would now frame a 2-year-old's existence, but it didn't frame mine. And I have this incredibly strong sensation of this recalled memory that is pure joy and delight as I'm running around as only a 2-year-old can. And there's little creatures of all sorts, grasshoppers, little butterflies, bees, I don't know, beetles, flying off in all directions. And just this sense of just sheer joy and delight about how alive it all was. And unfortunately, that was lost to me for many years, but taking a big leap to 1983, observing butterflies in particular first entered my life. By then I had a biology and ecology degree under my belt from the 1970s, and there was little work for anybody in that field other than becoming an educator or a librarian, and I've actually been both. So I was pretty active as an environmental activist from 1972. Well, I've never really stopped. and mostly I was working on sort of brown environmental issues, but somehow I was never doing the things I really loved to do. And it was mainly because I couldn't see the path. So I'll move my slides along. Oh, now, okay. So I'll just talk about a few formative experiences. So the lead up in 1973, freshly out of high school at that time, was during a camping trip to Lil Yabba Creek, and someone had come and had a light trap, and I was a girl from the burbs. I'd long since lost this exquisite sensation of being in nature, other than wanting to go on camps, and the creatures that came to the light that night just blew my mind. But the one that totally stood out was this Rotellia, if that's how you say it. Forgive me on my pronunciation of scientific names. It had an orange head and it was metallic blue-green, and it had these little tufts of white hairs on the abdomens, and there were little black bristles sticking out of them. And like, if you can fall in love with a fly, I did. It took me years to find out what genus the thing belonged to. This particular image probably isn't even the same species, and I found out later that their larvae are predators of aquatic snails. And around that same time, we did a group camping trip to Kulula National Park, and I got completely turned on to cockroaches. I know this is a little not butterflies, but because I saw this one and I could tell it was a cockroach because of the Circe and a few other features. And it was maroon with a translucent white edge all the way around the outside. I'm sure my description to myself is all the way around the outside. So I don't think this is the species either, but I'm pretty sure it's in this genus. And then there was one other occasion where a group of us were visiting the now drowned Texas caves near the New South Wales border. And I'd woken up early in the morning and I went to sit beside some little stream that was there. And there was just this sense of awe that a dragonfly nymph that had climbed up a reed and was proceeding to emerge and pump up its wings and harden them and fly off. I just remember sitting there transfixed. And these are all experiences that are so much harder to have now, and especially the ones that are sort of more at ground level of being a 2-year-old. So having acquired some degree of some knowledge about nature and the environment from a biology and ecology degree, I actually came away with very little understanding of anything, especially anything about life on Earth. But the longer I'm working with and around biodiversity, the more amazed I'm becoming at the intricacy and complexity and exquisiteness of our living world. It's just, yeah, anyway, it just keeps blowing my mind with each new story. So back to 1983, 2 things happened that were really memorable. And one of them was I gave my very first talk about nature conservation to a Toastmasters club. And it was called, "We've Saved the Whale, Now It's Time to Save the Snail." So you can see that my heart was already there, even if my actions weren't. And it was based on a New Scientist article about Polynesian snails that were down to a population of about 7. So at that point, I was becoming aware that the creatures that I loved so much were under some fairly serious threat. That year was also the year I gained access to a list of host plants for butterfly caterpillars. And at last, I had a way of directly engaging with the small creatures, at least with butterflies and their caterpillars. So my curiosity about small creatures got really reawakened in a big way. Up until that time, even doing a degree, I was really quite unconscious of the implications that insect herbivores were likely to be specialized to the host plants that they lived on most of the time. And while we all sort of knew that there were lots of invertebrates and there were way less things with backbones, nobody I know had any inkling of the sheer diversity of invertebrates. So of all the named species, they are 95, 19 out of every 20 named animal on the earth at this point, back in 2020, is an invertebrate, and we don't know how many invertebrates there are, so it's still a skewed data. So they only ever really receive attention even now, most of the time, if they're pests or if they're helpful to us. So is it beneficials or pests? And the story is just so much bigger than that. So with a list of host plants in hand, and I spent a few years living overseas in Holland, coming back in 1986 with a friend who was also interested, he set about outsourcing all the different plants that were on the list, and I started document life cycles of the plants that the plant supported. So at that point, my journey into observing butterflies had started in earnest. So let's, okay, wrong way, sorry folks. Okay, so my very first direct experience that year was while sitting in my garden and watching a blue triangle butterfly. And I didn't realize it was a she, but she sort of landed on one plant quickly, flew off somewhere else into the garden, landed on another one. She did this working her way up towards the front where I was, the front of the backyard. And then she found a cinnamon. I had a small cinnamon tree seedling. I don't know if you can see the size, with a flush of new, beautiful pink growth, non-native, but these butterflies like a few non-native laurels. She hung onto the leaves. She kept flapping her wings. She curled her abdomen round and laid an egg. And not that this is a photo of the egg, but down in the bottom middle is an egg of a little round egg of a blue triangle. So the image here is shows a caterpillar and a chrysalis, and the inside and outside wings of a blue triangle. Unfortunately, that particular one flew off too quickly to get a photo, but it was my first observation of egg laying on a host plant, and I raised that caterpillar, saw its chrysalis, missed all the stages. All of these images are much more recent, like from much more recent raisings of caterpillars. And it would take a little more time before I got my eye in. So many stages, and some of them go quickly. So I needed to learn how to see it. Notice when an egg's about to hatch, caterpillars are about to shed their skins, about to pupate, and when they're going to eclose, as it's called. I've raised, I don't know, because I'm really lousy at keeping records, but I've raised at least 1,500, if not 2,000 or more caterpillars, covering 75 species. And now I can see the transitions quite easily. And it's really worthwhile learning to see these things. It never grows old. So at that time, the only film that you could get was basically camera film. I was using 400 ASA Kodak Ektachrome film. It was very expensive to develop. In the intervening years from the 80s to the mid 2000s, I've probably got a collection of 10,000 slides that only a few have ever been scanned, because they're actually impossibly grainy, which is very sad. And I've basically had to redo all that work. I'm still redoing it. But now with the advent of inexpensive digital photography, we can all take as many photos as we want, which is great, because that's really fostering citizen science through platforms like iNaturalist. Some months later, I would take regular morning walks around a big block in West End, and it took me past a row of Cassia fistulas, golden rain trees that were street trees, and I'd see all this butterfly action, and they'd be paying, the butterflies would be paying close attention to all the little fresh shoots, which were mostly high up, so they were hard to see. And these new shoots were showing good signs of having fewer and fewer young leaves. So it piqued my curiosity, and I spent some time trying to figure out what was happening. It turned out, well, the butterflies were lemon migrants, which also eat a range of non-native plants, but have plenty of their natives. So well, the sequence of photos I've got here are of a black jezebel. It's also in the same family as lemon migrants, so the shape of the eggs are much the same, and the hatching process isn't too dissimilar. So there's them first starting. Oops. What did? So they come, they hatch. So you see little brown heads in the egg still. They chew their way out of the eggs. Lots of whites eat their eggs. And then that was the batch, and that's the black Jezebel butterfly that they were on their way to becoming, and a few of them became those photos. So the main difference is that lemon migrants lay their eggs singly rather than in clusters. It was a few days into observing the cassia fistulas that I did start seeing some larger caterpillars, and they were really well camouflaged, and I would bring those twigs home to raise them along with some extra leaves and other twigs. But every time I did that, I would bring, I started again. So after a few days, some of the bigger caterpillars that I had brought home, because they're only caterpillars for about a week, they'd started pupating, and you could see them then, but they were bigger, and my curiosity had told me that that's what they were doing. But very unexpectedly, every time I collected more leaves, I was bringing home more mouths to feed, because the butterflies were still laying, even though there was very little food left. And so the walks kept being, I ended up having to walk twice daily to get enough food. So on the right-hand side, there's a little tiny caterpillar on the mid-vein of a leaf. Hopefully you can see that. There's a rather oversized egg on the left-hand side with the head of a bigger caterpillar, which is probably going to turn that egg into a meal. And then there's still a very young caterpillar, much larger. That would be like 5 millimeters when I took that photo. So here's a not full sequence of them pupating. So the image at the top, shedding their skins. So they anchor themselves by their tails, and I've learnt, one of the transitions that I learned to see is that when the head starts looking kind of tight and small compared to the rest of the body, they're in the process of dropping their head capsule. So in the photo on the left-hand side, I get my lefts and rights mixed up, the caterpillar has crawled its way out of the skin. So if, at any stage, if you move the caterpillars while they're in this waiting to pupa, to molt, then they lose that anchor. And I have tried taking the skins off them, but it's very hard to do. Mostly they just end up dying. And then the last, on the right-hand side down the bottom, is one that's then that same caterpillar, and it's pumped itself up a bit to give itself room to grow. So they need to shed their skins 4 or 5 times to before they're big enough. So yeah, so just losing my way. So the thing that really flummoxed me about this was all the caterpillars, and it was several hundred at least of these, or my memory is that many, from the cassia fistula trees. All of them had been green with a black and white stripe. And so I spent many an intervening year, whenever I was doing a talk, telling people, you know, you've got a lemon migrant caterpillar if it's green with a black and white stripe. But then back in 2015, started doing some walks in a different part of West End, and there were some different street trees planted by the council, non-natives, but quite attractive to lemon migrants. And I ended up with them with no stripes and with little tiny dots that looked like they were yellow migrant caterpillars, because they have little spots rather than a stripe, and then one that was coppery and like. So my question became, if my perception could be so blinkered, and how was my perception blinkered more generally? And it's a good question to start using for nature observation. How our human cognitive biases blinker us is another whole story, but it's well worth looking at. There's a lot of food in there for how to do our messaging, I think. But within a week or two of these morning walks, there were so many butterflies laying their eggs on the cassia fistulas. And every single time I tried to bring in plants to take home for the caterpillars I had, I was taking the food from other caterpillars that had already claimed that particular spot. So it was, I've done population ecology and dynamic subjects, but this was like a really grounding experience for that. So I'll just take you through a sequence of a chrysalis caterpillars pupating. So I didn't catch this one in the act of doing the pupating. Lemon migrants are in the family Piridae and swallowtails in the family Pilionidae. Both attach themselves by their tails and by a silk sling, which I hope you can see. My screen's rather dark, so. And so this one has been, it's been developing its pupal skin underneath its larval skin. So these take 7 to 10 days, usually about 7 days as a caterpillar. And in summer, this whole process of getting to this point is like a few hours, like if it's a few hours, you're lucky. So here it's starting to, the skin, forgive me, the skin's starting to actually slough out of even the its breathing tubes, the spiracles along the side. This is meant as a stop motion animation, but I have to get there yet. And you'll see the pupal skin on the right-hand side emerging from the larval skin. So, and it wriggles and squirms and wriggles and squirms, and oh, it's just amazing to watch. So you've got a very potted history of probably a good half hour's worth of image taking. So here it is with its very fresh, soft pupal skin, and they take a little while to compact down into their pupal color and size. So that's a yellow color form, and then that's a brownish mottly colored form of that particular chrysalis. So I'll take you through it emerging as an adult, closing. So here you'll see that you can see the wings through the pupal skin. At this point, the adult skin has separated from the caterpillar, from the pupal, case, and it goes opaque and a bit powdery looking. And the abdominal segments, my cue for it is if the abdominal segments are starting to stretch out, then it's ready to come out. But one did film me once and sat in that condition for 2 hours before it came out. So here she comes, or he. So, and then, come on, screen. And this particular species is really challenging for new butterfly observers because it comes in 9 different color forms. Some of them are sex related. I haven't managed to get my head around which color forms belong to which inside color forms connect with which outside ones, but you get, yeah, anyway. So the main thing with this species is you learn to look at the flight pattern, and they're often a bit higher up, and there's a word for just what's the feel of the animal, and it's a little bit different from the other butterflies in the migrant group, because there's lemon migrants, orange migrants, and a few others. So the white migrant, but it's smaller, but the others are the same size as that. So my experience with lemon migrants was quickly followed by tailed empress. And unfortunately, well, mostly you don't notice this butterfly because it's a really fast high flyer going, doesn't go with the sound effects normally. And you can't see it. But that particular year, there was a very big population of them, and it's another one. Being in a deeply urban environment, I was finding the butterflies that could live on non-native plants. So it does use Chinese elm, and there was a big one hanging over the fence of where I lived at the time, and I found these amazing caterpillars. They look, yeah, they've got crowns on their heads. They're just like stunning creatures. If the egg is laid on plants with an entire leaf, then they only end up with a few bands, and I've seen one photo of one with no bands. And if they're laid on plants with ferny-leafed wattles, they end up with black bands on all the segments. And if you disturb them, they open them up and flash this fluorescent pale blue at you. So it's the caterpillars are almost more fun than the adults, except especially when the adults are just doing the fast high flying trick. But these butterflies are like a tipple, and you can draw them down by mashing some beer with some bananas with some beer, or if you've got some sort of sap oozing out of any of your plants, then they'll come for this fermenting sap. So that we played with this pair for 3/4 of an hour before we sort of put them away on a branch to leave them to their mating. So this is a picture of my garden when I moved in. I'd been living, so it's 1987. It's also an excellent picture of a finger, so forgive me. It's the only picture I took because it was on slide film. I'd lived in the Netherlands in the mid-1980s, and I saw how devoid of life its cultural landscapes were. And I could see the writing on the wall for what was going to happen here, because it's a very densely populated country. And when I came home in 1986, it was with this clear intention and determination that hasn't stopped to keep working on urban biodiversity and keep it as high as possible. Fighting an uphill battle just at the moment, but I do live in hope. So the whole process of observing butterflies really started in earnest from this point onwards. It was a small workers' cottage on 16 perches in West End. It had been a strawberry farm since the 1880s, subdivided for housing in the 1890s. It and the local area, it was about as devoid of native vegetation other than like occasional little plants that it possibly could be by the time I bought it. And my friend and I had started developing the butterfly garden as quickly as we could. So our learning process started with what butterfly caterpillars eat which plants. And over about 10 years, about 90 different species of host plants were planted out of an estimated at least 360 for the McLean-McPherson overlap region, covering New South Wales, northern New South Wales and southeast Queensland. So, come on. Oh, you go flash, yep. So one of the advantages, I'm actually giving you a whole lots of little case studies of observations, and hopefully you're drawing your own learning from what that might mean for practice. One of the advantages of having butterfly host plants and native plants on tap is the new information that there is to be discovered, and there's just so much more to go. We were also growing bush food plants. This particular one we had was Milla Milla, Eleagnus trifoliata. It's actually a delicious bush food. It's delicious, it's delectable, not just edible. And this really weird looking caterpillar, so in the middle at the top, turned up eating the flowers. They were just little at first, so you couldn't really see them. So I raised them through found out very quickly that you had to keep them in separate containers because they're actually carnivorous, and they were also eating any chrysalises that had formed. I only had a small number. And in fact, lots of other species are also carnivorous if they're in any level of crowd. So it's the more different plants you've got of anything, but in this case butterflies, the more you can start observing other things happening and seeing how biodiversity in a backyard can increase. So I've recently found this particular caterpillar on tuckaroo flowers. That's a few years back, and that was from Woodford, and on brown currajong, on Commersonia bartriama. They were doing really well on the flowers of that from Holland Park. And both of those, they hadn't been recorded for at that time. So with this particular butterfly, you rarely find more than one or two, except if you've got locust trees in fruit, which isn't happening in Brisbane anymore because there aren't any frosts, but it's probably the most reliable host plant for this species that I know of, because you find several on them and you get them nearly every year. At least somebody out at Ipswich that keeps posting about them, and I've gone to see them, has shown me. So I once found it on acacia maideneye flowers, but that particular caterpillar didn't make it through, and I couldn't get any more flowers of that species because everywhere I'd looked, the flowers were all spent. I did try transferring it to another acacia with that sort of spike flower, but it didn't take to it. So in that case, I wasn't able to record a new host plant, but I do spend my time looking at every acacia maideneye since to see if there's any evidence of this butterfly chewing it. There are a few other blues, lycaenid butterflies that do use acacia flowers. So butterflies are probably the best known group of animals, and their host plants are the best known, but there's still so much more that we can find, we can learn. So as we pursued the goal of finding out about butterfly host plants, we worked on a book to encourage others to do the same, and it was called Butterfly Magic. And the booklet did really well for being 24 pages, covering 12 butterflies and a few related topics. And at the time, it sold 1,600 copies pre-social media in the 1990s, which was a pretty good run, really, for a fairly unknown topic. With that booklet and displays of butterfly host plants in pots, I attended lots of community functions and had stalls, and collected all the contacts I needed to set up the Butterfly and Other Invertebrates Club in 1994. And through that group, I and others organised many field trips, and the club grew to about 600 people, members. And all of this was done pre-social media and what we call citizen science. So I was that president of that club for 12 years until 2006. And around that time, I was diverting my attention to doing a lot of work at Woodford Folk Festival site, Woodfordia, as we call it. The Butterfly and Other Invertebrates Club, I think, has played a significant role in citizen science, science communication, and community education about butterflies and other creatures. So before there were major advances in computer technologies from the 1980s onwards, the main vehicle for citizen science were organisations and individuals that produced newsletters and magazines, and the other strategies were book publishing and public speaking. So these are two books that one I co-authored, Create More Butterflies, in 2005, and produced and published, and the other was on keeping Australian stingless bees. And both of those have been very successful. They've sold between them 12,000 copies. So I think that's helped spread the word a bit, at least. But if you're making a start now, there's just so much more infrastructure available. So citizen science now has online platforms like iNaturalist. You set up an iNaturalist account, post the images you're taking of any animals that you know or you may not know them. It's becoming a major resource for understanding the distribution of different species and also for scientists that are working on particular topics. And if you want to, and it's a really good idea to start learning which family things are in, and but if you want to start to get an idea of your beastie before you put it up, then there's this plethora of Facebook pages that on all specific groups of invertebrates, as probably most here would know. But for moths and butterflies, I take my hat off for the work that Jackie Beer's Australian Butterflies and Moths page does. And there's many, many others, along with Boyk having a page, but Jackie's has just been attracting moths to a flame, you could say. So many people are turning up on it. And then there's other more general ones like, well, it was Amateur Entomology Australia, and it's had some but there's lots out there. So I've still continued with publishing and public speaking. I do a fair bit of public speaking. I do so badly want to stop the simplification of ecosystems and have us start appreciating the complexity and maintain as much of that biodiversity as possible. And I've worked on that to a much larger extent this time around with Dick Copeman to produce a book called Inviting Nature to Dinner, the benefits of bringing biodiversity to our backyards. In a way, this book actually charts my own development from simply growing host plants and recording different species and life cycles to I'm trying to lay out some of what I understand of the intricacies. It's just so mind-blowing. But more about that later, and but I'm running out of time. So in 2018, this is what my backyard looked like. It's now a 35-year-old host plant and wildlife garden, and there's about 50 existing host plants left on it. It's become a mini jungle, really. So I'm developing it as a garden for community and a community education space for gardening with nature. Because we've got this weird thing of we put all our plants in raised beds out of soil and out of plant communities. Like the more I think about it, the more I don't get it. But I'm trying to track where that particular behaviour has come from. But during this time, I moved my attention to a rather large activity called the Woodford Folk Festival that has a property out at Woodfordia at Stanmore near Woodford, and have been working on the butterfly and other invertebrates department. It's called the butterfly department on their scheme, and revegetating it rather more as an arboretum than as like a regional ecosystem. So my involvement with that, so this is a picture of me giving a talk. We used to have a whole bunch of volunteers when festivals were happening that would wear, I think, 5 different butterfly costumes that represented real species, because it drives me nuts when people make up butterflies when they're so amazing. And you can learn so much more if you actually work on something that's real, so you can learn about it. I've done a lot of light installation work, letting people see. We were calling it Creatures of the Night, and had a display of signs that I've produced at each festival and at the plantings, and running regular working bees, and the signs that I've made were often positioned beside the host plants. So that festival has a big presence, 20,000, or had a big presence. 20,000 people a day were coming through, and eventually there were about 14 volunteers associated with that department. And besides the 5 butterflies, eventually we had a dragonfly and a blue-banded bee, and I'm still hanging out to influence the situation, even though I've withdrawn substantially. So to get a beetle, because the folds of those wings are amazing. So I have actually pulled back, and I'm looking for people that are willing to take up the baton. And I'm happy to support them. So if you know anybody. So between the species I've raised in my garden, which I know is at least 46, and others from Woodford and the region, I've raised 75 species altogether, and a number of spiders and stick insects and beetles and a few sawflies, and yeah, so a range of things. Sorry, somebody's sending me messages if you're hearing a beep. So over time, my question evolved, and it's been evolving quite rapidly. I've been sitting with this question for a long time, into like which predators are specializing on which butterfly caterpillars, and how butterflies and other invertebrates support food webs. What role do butterflies and other herbivorous invertebrates play in ecosystems? So where are they and what are they doing? And around 2006, I started trying to get a database up. Now, you can't see this, but you can see all the connections. So this was a map of a relational database that I had wanted to see developed, but I became unwell for the best part of 10 years and couldn't pursue it very well at all, really. And it was a bit ahead of its time because the technology wasn't there to do it in any sort of scale like a naturalist can do now. And I quickly realized that to do it properly, it was a legacy project. It either needed institutional backing or it needed to set up a foundation, and my health wasn't up to that. But the technology is getting closer now, and I think we can actually start using something like iNaturalist to get the ball rolling on it. Because my take is that it's all very well observing animals, but unless you know what they're doing and how they're doing it and how they're interacting with everything, you're missing so much of the ecological complexity part of the story. And while iNaturalist is still, as far as I can tell, I have an account, but I'm not a user because, well, I just haven't had the time really. What I'm suggesting to people is that we can use it in a particular way. And as I do my talks and try to really expound on the awesomeness of our magnificent biodiversity and our biosphere, and as it becomes better known, I find people get quite overwhelmed because there's simply too many species to get a handle on. So I've taken to suggesting some time ago that we concentrate on one or two native plants, and this is the conversation I was having with Sandra about how to start this ball rolling. Concentrate on one or two plants that we have access to day, night, early morning, whenever, and start recording whatever we can observe that's using or happening on that plant and how it's happening. And to post those images, either already pre-ID'd or partly ID'd, and the host plant onto your iNaturalist account, and make sure that they are cross-referenced in the notes section with information about what's happening. Because that means in time that we can start collecting that relational data, and also likely someone will be clever enough to work out how to extract it. So I'm hoping that can work, but it does need the cross-referencing. And I was thinking that I've been suggesting when I've spoken to groups is that people pick different plants and then they can have a conversation with each other about what they're observing on the plants that they're working on. And that would be a way of using a community development and community education approach to connect and share information. So, but I'll start the ball rolling with Alphatonia excelsa. I've been talking for ages about wanting to do a short series on a particular plant of everything that I know about it and can find out about it that the plant supports. And so, and every time I've mentioned that I want to do this, I have not yet managed to do it because I don't quite have the skill set. So if anybody would like to support me with that, that would be fantastic. So everywhere I go, Alphatonia excelsa is soap tree or red ash is incredibly well chewed. Every now and then you find one that's not. It seems like it's a veritable all-you-can-eat restaurant for insect species. And my interest was originally piqued by the small green banded blue, which I've raised on it several times, and photos are the result. It's a gorgeous little butterfly. And I find the caterpillars really quite cute because I like things that are very subtle as well. They're so well camouflaged. It's a fun game to take people on a walk and be looking at what chew marks can you see, realizing that each species has its own way of using plants, or each group of species does, and looking for evidence that the creature is there. So I've collected a few personal observations. There's a little looper moth that I raised on it, Caspia rectaria. I have downloaded some other people's images. I have been crediting them. So there's this weird little moth. I'm not even going to try and pronounce some of these names. There's a beautiful gum moth that lives on it. I have not seen its larvae on it, and a galling insect that I see quite often. And then I've raised but never managed to get them to pupate this Phylloxonatrix. It's called the bramble sawfly. For some reason, I haven't worked out how to get them to pupate. They become a very interesting little sawfly, but it supports another sawfly that I haven't seen on it yet. You often see it with 3 rows of ringbarks, and that's a longicorn beetle that lays its egg above the ringbarks, and then their grub is provided with more food, because I can never remember which is the xylem and the phloem in a plant, but anyway, it's interrupting the flow and providing more food for the beetle grub. And I have a very strong passion for mistletoes, but a rather shallow level of information about them because, well, they're amazing. And it was shown that if you remove the mistletoes from a particular piece of bush, it's work done down south, the bird species diversity dropped by 40%. Some mistletoes feed so many different insects, but I'll just do one quickly, which is the Amiema congenita. But the Alphatonia supports 2, 4, 6, about 10, I can't count that fast, mistletoes, different species over its range. Some of them are known host plants, some aren't, but that could be a lack of recording. The Amiema congenita supports the black jezebel, which I've shown there, and we saw the little eggs hatching. The imperial jezebel, which I've yet to see around Brisbane, and 7 species of azures and other blues, including, sorry, I can't remember. And it's so well eaten, it's supporting a smorgasbord of other creatures. So to wrap it up, why is this also important? Why do we need to be mapping the interactions and doing citizen science? For every named animal, invertebrates rule the world, the animal world. 19 out of every 20 is an invertebrate. 14 out of every 20 is an insect species. This is on the figures I could find. And 1 in 3 is a plant-eating insect that's largely highly specialized. 3% of all herbivorous insects are generalists. Butterflies, not so much, but invertebrates on the whole, some butterflies are declining in numbers and species, and especially the ones that are reliant on undisturbed habitat, which is what we can do is reclaim disturbed habitat more easily and leave the bush alone. Invertebrates, I've developed a whole lot of a new food web and a whole lot of other information about how our view of food webs is completely skewed by our education system. Nobody's fault. And really 19 out of 20 animals on a food web should be one or other invertebrate if it was to be reasonably accurate. They're pollinators, and it's been shown that local native plants support a much higher diversity and usually higher number of insects than do non-natives, with a rare exception here and there of unusual plants. So they're really helping create healthy ecosystems and have so many other roles. It's just not funny. So I'll end with just putting up my how to contact me slide and open it for questions. And sorry, I was trying to keep it shorter.
Thank you so much, Helen. That was cool. I really enjoyed this presentation. And yeah, no, I don't think it was that long. Actually, we've still got 5 minutes and, you know, it's been really good. I've just been looking through the questions through the chat and I don't see many questions, but I'm sure people are still getting their head around all the information as well. How can
Sandra, we did have one.
Yeah.
One
Yes.
question we had was, how do you take such amazing photos? I for one am always trying to get them to be still. Are you just very, very patient, or what's your secret?
Nah. The reason I started raising butterfly caterpillars was to get good photos of them, because they sit still when they're freshly emerged. It's the only reason I started doing it, was because I was sick to death of trying to catch butterflies on the wing or out in the bush. I mean, I had a little bit of experience because I had those 3 examples that I mentioned, or 2 of, I had a few examples because they were close by, but it was really a case of, it's just easier to grow them and photograph them. Mind you, it isn't, because I've spent hours hand-holding cameras. I've got this whole sequence of common crow doing all its actions throughout its life cycle, or nearly all of them. And it, you know, sometimes I was up from 12 to 4 hand-holding a camera, and I'd turn my back to get a drink of water and the bloody thing would start doing what it was supposed to be doing. So like it, yeah, so that's sheer patience. I have learned a lot more patience doing this. But I'm actually using really crappy equipment. My digital camera is from 2006. The main trick I found was using a macro lens with extension rings, and it's not even got that many megapixels compared to what you can do now, and putting it at a very high what's ISO setting, so you get some depth of field. But I'm hanging out to earn some money so that I can actually buy some equipment that does photo stacking, because that's the technology now. But I wanted to say that if anyone has questions but don't have time, you're welcome to just send them through to me via email as well.
We do have some more questions. I just wanted to give Janine a voice as well, just because she's prepared a survey for all of us to fill out, so we can keep in touch and know what we're going to do post this presentation. So I'll just let Janine speak for now.
Sure.
Thanks, Sandra. And also maybe give people an opportunity to type in the chat, because we're not all fast typers. I just wanted to let everyone know, if you're looking for some follow-up information after tonight, the best way for you to get that is if you do fill out the feedback form that we'll send out at the end of this session. So the feedback form won't take you very long, probably just 1 or 2 minutes, and in there you can actually tick what you want to hear about, if you want to follow up on Helen's project, or when we're going to do another event like this or something like that. I also want to let you know, if you've been inspired, you're new to citizen science and you're thinking, well, what other projects are out there that you might be interested in joining, you can Google Citizen Science Project Finder, and you'll be able to find there's a website that is hosted, well, it's a partnership, but it's basically an Australian Citizen Science Association website. It's called Project Finder, and you can look through there and actually see what other citizen science projects are around. And I know that there are a few ACSA members out there in the crowd, which is great. If you're in Queensland, you're automatically a member of the Queensland chapter for ACSA. And so if you're wondering what we do as a committee, you're wondering how to connect with us, how to share your work that you're doing, just send us an email. You should have that through your ticket, and you can contact us that way. Thank you.
There are a few questions. Thanks, Janine. There's a few questions I might read out for Helen as well that have come up now. So what should gardeners do to encourage butterflies? Example, do's and don'ts. Are there any tips, Helen?
Oh, wow. If I was starting now, I would be replanting, I would be planting regional ecosystems that were either on site or nearby that were a bit drier. I wouldn't be doing what I've done. Once you've planted a plant, it's really hard to get rid of it, even if you don't want it anymore. I've discovered, I'm talking to all sorts of people about like changing their gardens, and you just become attached to a plant that you've grown, and it's happened to me too. So but yeah, try and do regional ecosystem gardening or nearby, because we're drying out. There's not a lot of do's and don'ts because there's still so, particularly for native plants, the growing conditions aren't that well known. I did a toilet block planting back in 2013 as a project around Woodfordia. Sorry, Woodfordia context is important. And I planted a Southeast Queensland native, Senna gordii, to start teaching people that there are native sennas and it's not all about Easter cassia, and we can replace Easter cassia with this beautiful native and several other sennas. And normally it scaffolds its way through the trees or the vegetation that it lives in, and it's called a scrambler. And I had scaffolding trees planted, but these plants just went, thank you very much, went whoosh, became huge. So what I'm saying is, no matter what you think you know, you probably don't know what the right thing to do is. So I'm not sure that that's a really good answer, but I'm a firm believer in suck it and see. But I would really recommend that you think about trying to reintroduce what species were originally present, unless your land is really substantially modified and it can't, but then you can look for nearby that could. Because I've become aware of just how intense all the interactions are in habitat, and we're really prone to picking winners, which I've done all my life. I'm trying not to do it anymore. I pick butterflies. If somebody had given me a list of beetle host plants, I would have picked beetles because I really love beetles. But yeah, our perception just tricks us all the time. So the do's and don'ts is always ask more questions is the do's. Try to see what the layers are. It just becomes just more and more fascinating as well. Like if it isn't obvious to how wriggly I am, it just becomes just more and more intensely amazing. Whereas if you're just planting straight for butterflies, it's a good place. That's where I started. It's a good place to start, but you can speed it up for yourself. You can learn from me and ramp it much faster. I hope that was useful enough answer.
Yeah, biodiversity, hey, is the key.
It's all about biodiversity. We need to just bring it back wherever we can.
Yeah. Helen, another question Angela has is, how do you actually raise caterpillars? I mean, do you snap off a branch and bring them back in where you can watch them? Because she's hadn't had any luck with that.
You can do that. I keep an awful lot of them, small numbers, in takeaway food containers. I don't punch holes. I just stand them on their side. I leave the bottom open so that the carbon dioxide falls out. And I open them regularly to change the leaves or to mist them if it's needed. So I raise an awful lot of caterpillars like that. You can't do that with tailed emperors. I killed like probably 20 before I realized that they needed to live on live plant. They build themselves a silk pad that they keep coming back to. So anytime you change the leaves, you make them just keep working, building silk pads, and eventually they run out of energy. And you actually, my experience of them is you had to keep them in some amount of sunlight or warmth. And I think I've noticed that with a few others, but it's really a case of seeing how fast they're going, because they really are sort of environmental energy power, like heat powered. So, but yeah, so I am planning to work with somebody soon on how to raise caterpillars, because there's a lot of people doing it now, but that would still take a little bit of time. But I have got a section in Create More Butterflies that describes it to some extent. So if you contact me, I can send you through those pages. Or better still, you could buy the book. But that keeps me able to pay off my house and garden. But anyway, sorry. But I'm happy to share that.
That's a great answer. I guess they're all very individuals, these little creatures, and they all have different.
There's a whole lot that are very simple. If you do them in takeaway containers, I put a sheet of damp tissue under them, especially if they're eggs. You start seeing the frass from the caterpillar, so you know the eggs have hatched, because some of them are so tiny. My eyesight's getting worse, so I don't see them nearly as easily anymore. Yeah, so there's a bunch of tricks, and I need to write it up because it needs to be part of this forthcoming book that we're doing. So, which I have said we would start doing, but there's always a delay.
Yep, so we have another question from Louise. Endangered butterflies, such as the Richmond birdwing and the loss of the birdwing butterfly, can we slow that decline?
Oh, it's already significantly slowed because it's been taken from vulnerable down. It's been taken down one notch on the endangered species list. I can't remember from what to what. The issue with that is it's not a butterfly that you can easily do in a lot of situations unless you're in or near habitat in the first place. Because I've just had conversations with so many people that have put in the plants. I've done it myself. I'm up on a ridge. I might have been lucky enough to attract one or two every now and then, but that's not supporting a population. And my garden is, I describe it as being for FIFO butterflies, fly in, fly out. I can't maintain a colony on the whole for any length of time. I did maintain a colony over winter last year of one of the pearl whites. I haven't had it ID'd yet because I think it's one from North Queensland that's made it south, but it doesn't match with any of the local ones. But yeah, so it's worth planting it if you're in the right places for the butterfly to live. And yes, but I also started some of this because I put a lot of work into a recovery plan for the laced fritillary, which in the end didn't get funded, and the butterfly is likely to be now extinct. There's still some question, but it hasn't been sighted in over 20 years. And any sightings now are always at a distance, and nobody that's done any work on it is going to believe it unless it's actually a pin specimen handed into a museum, which is very sad to say. But yeah, so for some of the more endangered butterflies, it's very much more about habitat conservation. And that is substantially the case for the birdwing, though you can, by planting them in the right conditions with enough, they're a creek-line plant naturally. So if you have those conditions, then the plant can thrive. Mine eventually just died. This is too dry and too high on a ridge. Does that answer? Yeah, so it's a case by case with a whole lot of these things.
Yep, thanks, Helen. Yeah, that's great. I'm just trying to look through some more questions. Yeah, lots of people are just having a discussion about different things, and
yeah, no, that's what I'd like to see.
Yeah.
There was one. Sorry, how do you work out how long moths and butterflies pupate for?
Oh, that can be so variable. So like the Wanda Brown butterfly has one life cycle a year. It's a little butterfly that doesn't come into Brisbane, even though I've got its plant. There's a lovely little grass called Oplis menis amulus, I think it is. It has one life cycle a year. The males come out, I'm told, I've seen males once or twice, 3 months before the females emerge. They mate and then are in the rest of their life cycle, their other life stages for the coming season until they emerge again. So that one, so some of them are known. A whole lot of what I call FIFO butterflies are quite fast. You'll have eggs for a few in summer. You'll have eggs for a few days, caterpillars for 7 to 10 days, maybe 2 weeks. Some of them do take a bit longer. You can often do several cycles a year. A lot of species are chrysalises for 7 to 10 days, again in the middle of summer, and then they emerge as butterflies. I can tell now when they're about to emerge, so I set them up so that they can emerge, pump up their wings and fly away if I don't need to take photos of them. But then there's others like the Fuscus swallowtail, related to the dainty and the orchard, and they all feed on introduced citrus, so it's a food plant we can share. With the Fuscus swallowtail, they've got this, I'm assuming it's a drought over bridging strategy. Most of the chrysalises will emerge quite quickly. I've only done them a few times because they're not very fond of citrus. They really like a more northern plant called Glycosmus trifoliata, I think it is, or pentaphylla. The name has changed a few times. A portion of them will just do diapause, stay chrysalises for a year. Another portion stay chrysalises for 2 years. And I did raise them once, and I had a chrysalis that lasted 2 years. And unfortunately, that's why I really wanted to do a book about it. If you've got those ones inside, they're not getting any environmental moisture.
Oh, I think we've stalled. Sorry, Helen, I think we may have lost you. Yep. Okay. Environmental moisture is the last I heard.
I might just stop Helen's share screen in case it's her
there she is.
Okay.
Yep, Helen's back, and I'll turn my video off as well.
I can't see myself.
Helen, could you maybe stop the share screen, because then it will save your bandwidth if it's staggering. That could be it.
Okay.
Great.
Okay.
Let's try that.
I can see myself.
Thank you.
Sure. Sorry about that. I don't do this very often. I mean, I don't do it on Zoom very often. So yeah, so the Fuscus swallowtail, if they haven't received environmental moisture and you've got them inside, then you need to mist them regularly, because the one that I did have emerged, it closed, as it's called, and its wings stayed crumpled. It didn't have enough moisture left to pump up its wings, which was very sad, especially since, well, for it, but also for me, because I still haven't got really good photos of that one. But yeah, so it is a case of, you can find out now really easily. When I started, it was much, much harder to find out. That's why I started the Butterfly and Other Invertebrates Club, because then you could collect people of all different knowledge levels, and some would know something and others would know other things. But now, you know, you join a Facebook page, you post your picture and you ask a question, and most of them have got amazing people with amazing content knowledge answering on some of the specialist ones. So yeah, so if you're starting with any of them, I suspect a lot more moths do more diapause than I know about with butterfly caterpillars. It's a really important strategy. Blue triangles do it. They'll sit there for ages until the atmospheric conditions are right, and they actually colour up overnight. And the only reason I know that was I'm a night owl. I saw one colouring up at about after 2 o'clock in the morning, and it was a butterfly by 6 o'clock when I looked at it again. And it had been sitting there for several weeks. And I knew somebody, Bob Miller, was from the Sunshine Coast area, Landsborough. He's now passed away, very sadly. He was an amazing butterfly person. There's so many amazing ones. But he had 26 pupa around the lintel of his of a door frame, because he wanted to see what was going on. And they all just sat there for weeks. And then one time they all started colouring up, and they were all different ages that he'd had them for. But yeah, that was in the 1990s that I saw him do that. So yeah, so there's just, with all of this stuff, you can give a simple answer, but there's always extra layers, and it's all the extra layers that just make it just so much more exquisite and amazing. If I haven't said that before. I didn't
Helen,
see that.
there's a question about how can you tell if a pupa has died? And there's a follow-up question from someone else saying that their Fusca swallowtail has been in a chrysalis for a couple of months and was wondering whether it's ever too dry for them, and can you spray them? Yeah. So like two questions, whether you know if they die, and the spraying. Yeah.
Lightly misted is off, like fairly regularly. It'll only absorb the amount of water it needs. You really don't know how long it's going to stay there. So the other question was, how do you know that they've died? Well, the most obvious things is if the chrysalis case has broken open in some way. If it's had a tachinid fly, you've nearly always got this very neat round hole with a thread hanging out of it. That's the tachinid fly pupa leaving. It's a bit harder to tell with some of the little wasps, but often there's little holes. What I'm always looking for is, I'm a science fiction nerd, structural integrity. If your creature is healthy, there'll be something about it that, if you look at it closely, will tell you that the features will be more, not necessarily the pupil or caterpillar features, but the colorings will be distinct. Any butterfly caterpillar I've had that's been parasitized, I'll take monarchs for an example, all the bands become a bit blended and murky. So you're looking for, like it's not clear and neat and crisp. And if it doesn't color up, and if it was more differentiated in its markings and it becomes less differentiated, it's probably dead. But as far as I know, that you have to learn that by experience, unless you're hanging around with somebody that, or posting the pictures. Like I've seen plenty of, is this pupa dead on Australian Butterflies and Moths page. So, and it can be useful, because if I see it, then I'll say, well, you'll know it's dead when such and such has happened. If such and such a creature has made it food and it's become part of the food web before it became a butterfly, which a whole lot of them have to do. We would be in an impossible situation if most butterfly caterpillars and pupae didn't die. So,
because we have so many butterflies.
Sorry?
I just laughed because we would have so many butterflies. It would be an impossible situation.
Just a quick calculation, and this is just pure maths. One butterfly is said to lay, female butterfly is said to lay 400 eggs. If half of them are female, and it's usually skewed in favor of females, but just let's say we've got 200 female eggs and all of them survive. In the next generation, there's 200 by 200 females, which is 40,000. So there's not many. It's actually my Cassia fistula lemon migrant story, but done in numbers. But it wouldn't be just 40,000 caterpillars, it'd be 80,000 because of all the males. In the next generation, 40,000 by 40,000 is 1.6 million, which is actually 3.2 million caterpillars turning into butterflies. So the food sources would be just become non-existent. So everything is designed to be food for something else at some point in our lives. So, and butterfly.
And birds particularly, hey? Like ecologically, they feed a lot of other animals, like birds, for example, absolutely require the protein or other, some compounds from caterpillars. So it's interesting, as you say, the who eats who kind of scenario is so interesting and ecologically important.
Oh, sorry. I keep interrupting. Daryl Jones, Griffith Uni claims, and nobody's contradicting him as far as I know, that all Australian birds, irrespective of what they feed on themselves as adults, feed their nestlings insects. So if we're saying what's happening to the birds, you can bet your bottom dollar it's because we're wiping out the insects, and we're wiping them out at ground cover level on broad scale in agricultural systems. And we're taking over more and more systems for agriculture. So, which is why we really need to bring back as much biodiversity as possible. So, yeah, I just find that connection to birds really important. And yeah, there's some, we actually need to figure out which birds specialize in which groups of insects, I think, but I haven't managed to work out how to do that yet. So it's not about
lots of citizen science to be had.
Sorry?
I just said lots of citizen science to be had, as in we all need to, you know, become the scientists and help out.
Yep. There is a handbook of Australian
questions.
and New Zealand birds apparently that does have some information about what different species eat, but I haven't had a chance to get my hands on it, and I haven't had the time to. I'm rather constrained in action at the moment. So we're
well, look, Helen, thank you. Yeah, we're over time, but it's been so awesome again, once again. Every time I hear you speak, there's new light bulbs going off, and you can, you know, just we can chat forever about this.
Yeah, you and I can talk the week off.
You and I, and probably many, and and many people I can see a copy of that. Thank you so much, everyone, for coming and for being here to support Helen's wonderful work and supporting ACSA and communicating with us and having us in your living room or wherever you are in your bedroom for tonight. And yeah, thanks so much, Helen. We'll be in touch again with you. And if anyone's interested in trying to come up with, you know, if anyone wants to do the project, as in let's do what I'm saying, let's get this going off the ground, because I think it's a wonderful new way of looking at biodiversity through the ecological relationships. So thanks so much. And if anyone else has anything to say, I'll let you do that now, and then we'll say our good nights.
And I'd like to say thank you to everybody for coming. Much appreciated.
Thank you, Helen, and thanks, Sandra, as well.
It's wonderful, Helen. You haven't seen the chat, but it's just been fantastic. Yeah. It's been fantastically received. So thank you.
Because it feeds all the work I'm doing at, well, further communication and potentially other books that I've got a whole bunch of books lined up in my head waiting to just find the time to come out.
Excellent.
Do you know of any butterfly groups in Gold Coast or Northern New South Wales at all, Helen?
Not offhand, but I see new Facebook groups starting up all the time. It's such a different world from the one I grew up in and have done most of my action in. And I was an early adopter of technology, but it is passing me by to some extent. So much of it is just happening on Facebook now. So I know there's a Black Oil Range group, and I've seen other ones turn up, but after a while, I just concentrate on ones that are larger, because if they're in your region, you can swap notes with people from your region by keeping track of the posts that are there. I did, in starting up the Butterfly Club, I did try to get branches to form so there would be more regionalisation, because there were lots of people from all over, or at least sort of southeast Queensland, and northern New South Wales, but that just didn't happen. So, and it's too late now because technology is just working we operate differently. But yeah, but I'm pretty sure if you, VoIC is one, and then Facebook pages.
Great, thank you very much.
There are people on the Gold Coast, there are people on the Gold Coast that are active, because there's a butterfly garden or butterfly house down there, and I hear about people like that, and I know individuals, but I haven't heard that they're meeting as a group. Sorry, I'm just trying to think up my answer.
I just thought of something as well. Even a naturalist kind of scene, when we start uploading this data that people will start collecting. I wonder whether we could do a naturalist project, when you put up your photo and your description and everything, but also you can feed it to a project. So we could literally create a project. And I think that would be a useful thing. So the data goes through the project, and therefore we can manage the data as well and understand it better and interact with it. So maybe that's one thing where we can actually create a project where people also will, you know, mark that they're adding this particular observation to the project. That's one other way we could run it.
But would need to have the direction that you need to be recording the plant and the animal.
Yes.
And cross-referencing it. That's the bit that
Yes.
I don't know how to feed that into the system, but it needs to be fed into the system.
I can help you with that. We will talk later.
Okay, happily
Excellent.
talk to you more about that, because yeah, well, hopefully you'll see how useful that would be.
Mhm. Perhaps for another one, one of these, Sandra
Yes.
and Janine, we could do
Tom,
an iNaturalist one. I can ask a couple of friends of mine to help out with that. Would
I've
anyone
been
else
dreaming of
be?
that.
Would anyone else be interested in that? If so, give us a yes in the chat. That'd be great.
Yay!
All right.
Let's get Thomas in if we can.
I'll ask him.
And invite me so I can chew his ear off about the relational databases.
I love that.
Then he can up into the system.
Mm-hmm. So there is a couple on the Great Southern BioBlitz YouTube channel, if you do want to have a look at those.
Yeah.
He goes very much in depth. They are quite a long session, but I suggest you join into the first sort of half hour of it, and you'll see most of it there. He doesn't go into so much how to use the app, and that's a whole nother ball game, but I can do that one too. Absolutely, Lynn, if you would like, I will stick the website in here for the Great Southern BioBlitz YouTube channel. And just give me 2 seconds. Switch counts, GSB.
Are we allowed to turn this off if Jen leaves? Because I'm just seeing that Jen really needs to go and we're still going and she's the host. So are we as hosts able to leave
I don't
without Jen?
think so. That's the problem.
Okay.
Anyway, here we go. I'll just put this in the chat now. If anyone wants to just grab it quickly. That's the list of how to use iNaturalist from the Beachcomber from Great Southern BioBlitz. So that's a really, really good resource to have a look at. There's a number of little how-to videos or long how-to videos on there. But yeah, stay tuned on that score and we'll see what we can do. And yes,
thanks so much, everyone. Yeah, that's awesome.
And thank you to the people that have made it possible. All of you.
Thanks, team. You've been great. We better finish, so we'll let Jen turn us off. Thank you so much, everyone. It's been a wonderful first go. Thanks, Helen.
Everyone. Good night.
Bye.
Night.